Hey, good morning, everyone. Just a quick reminder as we start this week. Next week is our second exam. Hopefully, all of you were already aware of that, and it doesn't come as a shock to you. But we will talk a little bit more about it later this week. But keep in mind, this week in recitation, you will have your homework due from last week. You'll have a quiz, and it'll be the last time you'll have your recitation meeting with your TI before next week's exam. Right. So I think some of your TIs may hold optional review sessions, which is great. And of course, I think it's wonderful if you take advantage of those. But just keep in mind that that's where we are headed as we start this week. Let's shift our focus and learn about the importance of the membranes that make up our cells. And what we've discussed membranes in terms of their composition. Last time we looked at how we have this fluid mosaic model of the membrane, and we began to appreciate how they are composed of different types of lipid molecules, like the phospholipids, as well as different types of proteins that are all embedded in the membrane. And that composition of lipids and proteins is a very important tool for how our cell can regulate the passage of things in and out. Oftentimes, we think of cells as being just kind of membrane-bound sacks of proteins and nucleic acids and carbohydrates, things like that. And we don't really give a lot of credit or pay much attention to how complicated the processes are that take place right at the membrane itself, and how the membrane cannot just be sort of a, permanent physical barrier against everything, that very clearly things must be allowed to pass from one side to the other in order to allow for different life processes to take place inside the cell. So this lecture and the next, we will look at different examples of how things are regulated, moving across these membranes. So today's lecture will introduce this and talk about simple and facilitated diffusion. So let's start off by just getting on the same page, how to think about molecules that are floating around in some sort of a solvent, right? So little molecules that are mixed in some sort of a solution. The little molecules themselves would be the solute, right? And then whatever type of solution they are mixed in would be the solvent. And the most important solvent when we're talking about living things and cells is, of course, water, right? Water is the most important solvent when it comes to life here on Earth. We've all done this before, where you have some food coloring and you drop it into water, and this food coloring over time will naturally spread through the water. But have you ever really thought about what is going on there? I mean, this is one of these things where it's so intuitive that ever since you're a little kid, if you drop some color in water, it'll spread out. But why does it do that, right? Why doesn't it just stay as a droplet in the water? That's because water, again, has these remarkable properties of being a polar molecule that interacts with itself, as well as with different types of things that are put into it. And those different molecules that are put into it, depending on if they are hydrophilic or hydrophobic, that will have a big impact on how they behave when they're put into water. Let's look at this little illustration here. I think this is pretty obvious what this is depicting, that if you were to put a concentration of a particular molecule in something like water, right? If this is a hydrophilic molecule, meaning that the water molecules will very happily surround it, and through hydrogen bonding, right? Each one of these little black, what are these? 1, 2, 3, 4, 5, 6 hexagon little molecules will be surrounded by water, right? And as a result, the water molecules, as they're bouncing around these, right, will naturally want to sort of separate these little molecules apart. right? Because if you think the water that's not pictured here, but it would be in the white space, right? Teeny tiny water molecules, they would get around each one of these, and the water molecules are all kind of vibrating and bouncing against each other. So very naturally, over time, right, these black hexagonal molecules would spread out evenly so that all the water molecules between them, right, there's no real spot here where the water molecules are more crammed together than any other spot. Right? So in other words, the spreading of this food color or these black hexagons is not so much the molecules of the little hexagons themselves wanting to spread out, right? It's more the water between them naturally spreading them out so that the water molecules are happily sort of evenly distributed among them. So that would be if it's hydrophilic, right? This particular food coloring. But what if the substance that you put in the water is hydrophobic? In that case, all the water molecules that are not pictured here in the white space, right, they don't want to interact very happily with these molecules. There's no hydrogen bonding, right? There's no charges that they would like. So as a result, the water molecules are just physically bumping up against them. And as a result, they would naturally cram all of these black hexagonal molecules together, right? Because in that sense, all the water molecules around them bouncing around would just naturally cause them to stay together, again, to minimize the amount of water that is unhappily sort of stuck between them, right? So that's why if you drop a droplet of oil, for example, into the water, right, the oil will not spread out. The oil will naturally be kept together, not because the oil molecules love each other, right? It's more because the water molecules keep them together. So this property then holds true if you have, let's say, a membrane that's dividing the top and bottom part of this chamber here. So here we have, again, some sort of substance dissolved in water, right? Or you could even imagine that in this case, these little blue things are picturing the water molecules themselves. Then if you have a membrane like a lipid bilayer, which again is held together because of these competing polar interactions of water liking one end of the phospholipid molecule, right? So happily attaching to those, but the fatty acid tails of the phospholipid, right, just get pushed away by the water. So naturally the tails of these phospholipid layers would point together into the middle. If you have water in here, water can certainly go through the lipid bilayer. And you say, what? I thought the whole thing was the middle part of the bilayer, is hydrophobic, right? Hydrophobic doesn't mean that water molecules cannot go through. Hydrophobic simply means that if you have a lot of water pushing on them, they will naturally be pushed to a place where there's low to little water molecules. So in this case, if you have a high concentration of water molecules on one side of the membrane, right, they will naturally want to spread out from each other as they're bouncing around. So some of them will go through the membrane, and over time, the concentration of water on the top and the bottom would be equaled out, right? Because in that state, then there's no real motivation for the water molecules to get apart from each other anymore, because by then they're all spread out so that no matter how they move, it's sort of like equal interactions with each other, right? But you would end up with much fewer water molecules in the middle of the membrane, right? And that's kind of the point here. So this could be water that we're talking about, or importantly, it could be some other type of biological molecule, right? Something that the cell needs to have come in, right? Or needs to have go out. So the point is, in this particular case, even if you have a membrane there, right, the whole principle of water or molecules wanting to spread out to even out applies. And it's interesting to think that this is nothing more than just a physical property, right? This has nothing to do with like, oh, life invented this or something like that, right? And I think all of you realize that, right? Because food color in a little flask of water, that has nothing to do with like the evolution of life, right? That's just a physical property of a substance in water. But because this physical property was there from the very beginning of life on Earth, as Earth was covered in liquid water, it makes sense that all of life processes that evolved, right, and to this day we still depend on, has to do with these properties of how molecules in water behave, especially across things like a membrane. So in our cells in our body, it's not a simple matter of just saying, oh yeah, stuff on one side of the membrane, if it's more concentrated, will always flow to the other side until they even out, right? Because even though that might be sort of the natural physical way that these molecules want to move, relying purely on that would not be compatible with life, right? I mean, think about it. If you have a living cell and the way the cell works is entirely dependent on how naturally molecules want to sort of go from one side to the other, you couldn't really have any life processes taking place, right? Life, the biochemistry of life, is all about concentrating things in certain places so that things can be encouraged to happen. So that means our living cell and its membrane is always coping with how to kind of address things wanting to move, things wanting to move out, right? As well as, considering that some things will just want to move based on concentrations of how many molecules you have on one side versus the other. But because a lot of biological molecules are charged, positive or negatively charged, the cell also has to deal with a concentration, not of physical molecules, but of charges, right? And I think you can also intuitively know that if you have one side of a membrane that's highly positively charged, right, and one other side that's highly negatively charged, then the charged molecules that create that separation will tend to want to move in the direction of charges to even out the plus and the minus. So as a result, our living cells are always dealing with that. The actual numbers of molecules on either side determining a gradient, as well as the distribution of charges on either side of the membrane, also causing a gradient. So in this cartoon here, this is sort of a nice introduction to thinking about the difference between right outside of a cell's membrane. So this blue area would be extracellular, right? And then this is the boundary of the cell's cell membrane. And then inside of the cell, this kind of peach colored area would be the intracellular region. And so if you look at different types of important biological molecules here, we can see certain ions, so like sodium ions, right? And you'll notice that sodium ions, right, tend to be higher concentrated outside of the cell compared to inside, the blue. So in other words, the natural gradient flow, right, of concentration of number of sodium ions would be from the left. It wants to go through the membrane to the right to even out the number of sodium ions. Potassium is sort of the reverse. You have way more potassium ions inside of the cell, right, compared to outside. So the natural flow in terms of number of potassium ions would be from the intracellular out to the extracellular. Okay. But keep in mind, right, that the cell is actually keeping these two concentrations on purpose imbalanced like this, right? And we will learn why that is, right? Because by keeping it imbalanced like this, right, you allow for controlled ability of these ions to flow in either direction. Keep in mind also that the inside of the cell has more proteins, right? Because obviously the cell's making a ton of different proteins, little machines inside of the cell, or proteins for structure. Proteins tend to be negatively charged. So as a result, the inside surface of the cell membrane tends to be more negatively charged because that's where you have a higher concentration of things like proteins, and the outside tends to be more positively charged. So you have more of these positively charged ions on the outside. Okay, so let's explore how the cell actually controls this. What we will do is over the course of the next few lectures, look at various examples of different membranes in the cell and how important proteins are involved at those membrane surfaces to allow for things like controlling the flow of molecules in order to do things like balance out different properties from one side of the membrane to the other, as well as utilizing the flow of molecules through the membrane to actually do some work. Right? So if you look at this cell here, this is just an example of many different membranes, right? So the outer membrane of the cell, we will look at examples of how we have certain proteins that help carry things like glucose from the outside of the cell into the cell, or help facilitate the flow of water from the outside to the inside of the cell. We will also look at certain organelles, like the surfaces of mitochondria over here, where you will have, this is zoomed in here, showing that you have particular proteins that will take advantage of gradients from one side of a membrane to the other to allow highly controlled movement of molecules across that can then be harnessed for energy. We will look at mitochondria. Later in the semester, we will look at chloroplasts and how chloroplasts have a similar system. And we will look at things like different vesicles that can move things in and out as part of doing things like packaging certain molecular products, like finished proteins that are coming from the endoplasmic reticulum that then need to be moved around inside of the cell, or maybe let out of the cell if this is something that the cell is meant to secrete. Right. All right, so let's take our look at this. So any situation where you have a membrane dividing two sides or two spaces or two volumes of a solution, you have a potential for a gradient to be set up. And so anytime you're moving molecules in one direction or the other, by definition, you're either moving molecules along the natural flow that they want to go in if they follow the gradient from high concentration down to low, right? Or by definition, if you go in the other direction against the gradient, right, you're moving molecules from low concentration to high concentration. So in this first slide here, let's look at some just basic examples of this. So here on the left are two examples where you're moving things passively, right? Passive means it doesn't require any energy because you're just utilizing the natural flow in the direction downhill of the gradient. So we said, for example, this is what we talked about before, these little blue molecules could be just some teeny tiny little biological molecule, or you could just say these are water molecules, for example, right? And if there's a higher concentration on the outside versus the inside, then these water molecules, or whatever these little blue things are, will naturally want to spread out. And some of them, if they're small enough, can just sort of work their way through the lipid bilayer and just get to the other side, right? So in this case, this is passive because the direction is just naturally based upon the physics of these molecules. They want to go from high to low, but the fact that there's a membrane in the middle sort of gets in the way, right? So the membrane would slow this down, right? Another example is, what if you have things that are on a high concentration on one side and low on the other, but this molecule over here, right, this red molecule here, maybe is too big to sort of just make its way through the lipid bilayer by itself, right? If you had a tunnel through the membrane, for example, this big green thing, right, is a tunnel that allows these red molecules to just flow through, then in this case, right, you are still passively letting them flow in the direction that they want to go in from high to low concentration, but you are helping them do it by providing a path through the membrane. So you are facilitating their diffusion, right? So these green things, what are they? Well, they would be proteins of some particular shape that's meant to allow this red molecule to go through it, but maybe not allow certain other molecules to go through it. Okay. Then this last example here of active movement or transport. So in this case, we're no longer talking about diffusion, right? Because diffusion, just like the drop of food coloring in water, diffusion is a passive process just having to do with concentration, right? In this case, this is no longer diffusion because you're going in the opposite direction. You're saying this purple square molecule, for whatever reason, right? The cell wants to get a lot of this into it, but there's very little of it on the outside, but whatever, the cell wants to take whatever it can find on the outside and bring it in, even though it already has a lot of it on the inside, right? So in this case, very obviously, the purple arrow is going against the natural diffusion gradient, right? It is going sort of uphill, if you want to think about it that way, right? And think about going uphill is a useful way of thinking about it because this requires energy to do it. So this big green protein here, whatever it is, right, won't just on its own move these purple things through. It's going to need some energy to actually allow it to change its shape against the direction that these purple squares might be trying to otherwise, force it to change. All right, so let's explore these diffusion concepts today, and then next time we'll look at these active transport concepts. If you're just talking about simple diffusion, meaning all you're talking about is a membrane, and on one side there's a high concentration of something, on the other side there's a low concentration, and whatever this something is can make its way through the membrane, even though the membrane kind of slows it down a little bit, right? You can get this type of relationship where as you increase the amount of solute on the x-axis here, starting from nothing to high, what are we talking about? We're talking about these little red molecules are the solute that's dissolved in a solvent like water. If these little red molecules can just diffuse through the membrane, but the membrane kind of slows it down, it makes sense then that the more solute you have, right, the rate of diffusion, meaning how quickly can these little red molecules get through the membrane, that doesn't change, right? Because it's not like just because you have more of the red molecules that they somehow get better or quicker going through the membrane. It just means that there are more of them. So the rate of diffusion will just increase based upon how many red molecules you have around there, right? So if you're measuring the rate of diffusion as how many of these little red molecules are actually making it across the membrane at a given time, right? It's a linear relationship. However, if you have a situation where, let's say, this green molecule, right, wants to flow from high to low across the membrane to spread out, but it has a hard time getting through the membrane by itself, but you have some sort of a protein, this blue thing here, right, embedded in the membrane that provides a pathway for this green molecule to just flow in. Right? Then you might imagine the relationship like this. As the concentration of the green molecule increases, right, they start being able to flow through these membrane pathways. Now, keep in mind that on the surface of the cell of this membrane, you wouldn't just have one of these blue molecules, right? You'd have a certain finite number of them available. So if you have very few green little balls arriving, right, then of course, if you have plenty of these blue diffusion proteins in the surface, then at first, yeah, there's nothing kind of slowing it down, right? There's plenty of these channels for the green things to get through. So at first, you have a very rapid rate of diffusion of these green molecules coming in. But at a certain point, what happens? As you increase the amount of these green little balls, these solvents, right, at a certain point, because you have a fixed number of these blue proteins that let them come in, at a certain point, right, there's way more green molecules out there than the blue little pathways can accommodate. And so you will sort of top out, right, at how quickly you're getting these molecules into the cell. So in other words, we say that you reach a saturation point, right, where no matter how much more of the green molecules you have, right, you don't have enough of the blue little channels here, proteins, to accommodate them. So it's kind of like a situation you think about like this, right? Like the Stan Sheriff Center on game night. The Stan Sheriff Center has many different entrances, gate A, B, C, etc., to let people in, right? So if there's no people on the outside trying to get in, then obviously you would have zero sort of rate of diffusion of people going in. But then as the gates open and you start having people collect on the outside, right? At first, the first few people, right, there's nothing to impede them from going in because there's, maybe like a dozen or so entrances to get in. Right? But at a certain point, more and more people start showing up, and then all the entrances have people flowing in, right? Once you get that certain point, boy, that's a great balance, right? There's plenty enough gates for people to go in, nothing's slowing it down. However, if you have more and more people showing up, in other words, the solute concentration gets higher and higher, at a certain point, the entrances to the Stan Sheriff Center are saturated, right? And no matter how many more people pile up on the outside, right, that rate of how many people are actually getting in at any given moment is limited by the number of gates there are. Right. So why is that concept important? Because much of how our cells function have to do with dealing with having enough gates or entrances to accommodate certain changes in concentration of solutes that want to be moved around. And importantly, being able to interfere with those gates that allow molecules to flow in, right, is a critical way that different types of cell functions can be controlled. So let's say, for example, here we have a membrane, lipid bilayer, and this dark purple protein is one of these, let's say, transport proteins. And this transport protein, on the outside of it, you can see it's the right shape. It's the right shape to bind this light blue turquoise protein, right? And so this light blue turquoise protein would normally sit in this nice kind of shaped region of the protein that's meant to hold onto it. Right? So this thing would buckle in here, and this part of this transport protein would really kind of hug the side here, so hold onto it, and then it would allow it to go into the cell. Right? So if we were just to think about the movement of this light blue protein into the cell based on the number, the fixed number of how many of these dark purple proteins are on the surface, it would follow this purple line over here. Right? So as the substrate levels increase, substrate referring to the light blue molecule that's being transported, right? And you were to follow the rate of the reaction of how the substrate is being handled by these transport proteins. Right? If nothing is getting in the way, then you have that very typical kinetic response of you reaching a saturation point, right? Where at a certain point you have so many of these light blue molecules that the rate of them moving in can't go any faster because it's limited by the fixed number of the dark purple receptors, right, or transport proteins. However, what if the three-dimensional shape of this transport protein not only is meant to bind this light blue turquoise molecule, but can also bind this dark blue one? And importantly, if it binds this dark blue one, then it's no longer the right shape, right? Because the combined shape of the purple plus the blue would no longer be the right shape to bind this turquoise one. Right? So that would be an inhibitor. In other words, this navy blue molecule is inhibiting the ability of this transport protein to move the turquoise one. Right? In this case, though, you can imagine it's an either or. At any given time, this purple receptor can bind the blue one or the turquoise one, but not both at the same time. And so it would take turns. If at a given moment, the dark blue one is bound, that prevents the turquoise one to bind. If the turquoise one binds, it prevents the dark blue one. Right? So that's why we would call this dark blue one a competitive inhibitor. Right? It is competing against the turquoise one for binding of this transport protein. Now, you might already think what I'm getting at is this competitive inhibitor might not be a natural product of your cells. It might be some sort of a medication or a drug that works this way to prevent your cells from taking up a particular compound or molecule, right? So for example, just a couple of examples, you have pain relievers that can interfere in a competitive way so that cells like your nerves, right, are less responsive to receiving different signals that are coming in because those medications block receptors in a competitive way. Right? Or allergy medications like antihistamines. They are the right shape to bind to the receptor proteins on the surfaces of cells that would normally be receiving histamine signals that cause inflammation and allergy reactions. And they do so competitively, right? That's why at a certain point, if you have too much pollen in the air, right, your allergy medication doesn't work. Right? And over time, the allergy medication is just eliminated by your body. So if we were to follow the kinetics of that, that would be the green thing here, right? Competitive inhibitor at work. Right? And we're still measuring, right, how much of the turquoise molecule gets moved into the cell. Right? So you can see because there's a competitive inhibitor present, right, the rate of transport of the turquoise molecule is kind of slowed down. Right? But at a certain point, if you have way more of the turquoise molecule than your competitive inhibitor, right? So as you move farther to the right here, at a certain point, the turquoise molecule outcompetes the inhibitor, and you still reach that same saturation point. What's particularly exciting then is if you can come up with a non-competitive inhibitor, meaning you have a molecule here that binds somewhere else on the transport protein or the receptor. And by doing so, right, it basically changes the shape of the binding site up here so that now, right, this turquoise molecule, yeah, it can still kind of fit, but it doesn't get held in place, so it just falls out. Right? And importantly, it doesn't matter how you adjust the levels of the turquoise molecule, just as long as you have enough non-competitive inhibitor to just change the shape of the receptor here, right? It doesn't matter how much of the turquoise you have. So that's what we call a non-competitive inhibitor. So as you increase the amount of substrate, at first, right, not all of the receptors are bound up by the non-competitive inhibitor, right? But at a certain point, right, they are bound up. So even if you drive higher levels of the turquoise molecule, right, you still get almost permanent inhibition of the uptake of the cell of this particular compound, the blue one. So many pharmaceutical drugs, the real goal is to design them as non-competitive inhibitors. So you can get away with just having a very low dose of them, and yet their effect on altering cell activity is, more effective. Okay. So let's kind of summarize this and think about this in terms of different types of requirements for these to happen. So we said that diffusion is just a way of defining when you have a relatively straightforward situation of things in a high concentration flowing through a membrane down to a low concentration. So if you think about a tea bag in hot water, the small molecules that, like the different tannins and things like that in the tea that give tea its color and its flavor, those can simply diffuse right through the paper of the tea bag. Right? The tea leaves, on the other hand, are too big to flow through the paper. Right? But if there are little tears in the paper, little holes in the paper, then the tea leaves can also flow out. And in either case, right, whether you're talking about the little tannin molecules and the flavor molecules or the tea leaves, they'd be moving in the direction from high concentration to low, right, from the tea bag out into the water. So the tea spreading is just simple diffusion. The tea leaves spreading out of the tea bag will require certain openings. So this would be facilitated diffusion. But again, doesn't require any work. However, the opposite, if you were to imagine taking all of the spilled tea leaves that might have leaked out and all of the tea molecules that are in your tea here and somehow getting them back into the tea bag, that obviously won't naturally happen as a result of these molecules behaving, just by physics alone, right? This would require energy. You would need to have specialized molecules to do this work, to move them up against the gradient. So let's compare in this chart below, simple diffusion, facilitated diffusion, and then we'll talk a little bit about active transport, even though we'll focus more on active transport next time. So in the case of simple diffusion, some examples of solutes that are transported just by simply diffusing, things like small polar molecules like water, glycerol, right? Small nonpolar molecules like gases like oxygen, CO2, they just simply diffuse across membranes. And large nonpolar molecules that tend to be hydrophobic, things like oils and steroids, right? So different types of lipids, they will move through a lipid bilayer very easily, right? Because they have many of the same properties as the lipid bilayer itself. Right? So in terms of thermodynamic properties, the direction relative to the electrochemical gradient, they're all going downhill from high to low. Is there any metabolic energy required to do this? No. And there is no intrinsic directionality, meaning the membrane is not designed to only allow things to flow one direction, right? It could just be that once the gradients shift, these things will flow in the opposite direction. So there's no real built-in defined directionality. In terms of kinetic properties, would this simple diffusion require any special membrane proteins to help them out? No, right? So therefore, there would be no saturation kinetics of proteins helping these things move across, and there'd be no opportunity for things like competitive inhibition to happen. For facilitated diffusion, so this could also involve small polar molecules like water. So as we will see as an example in a couple slides, if certain cells need to have water move through the membrane faster than simple diffusion allows, it can do so with the help of certain proteins to facilitate water movement. Large polar molecules like glucose, right, depend upon facilitated diffusion to be moved over with the help of proteins. Certain ions like sodium, potassium, calcium. So let's look at these traits down here. Because it's still diffusion, right, these molecules are still moving down their natural up-down chemical gradient, right, high to low. So no energy is required, and there is no intrinsic directionality of which way they need to go, right? But the difference compared to simple diffusion is down here. Here, in facilitated diffusion, you're going to need to have some sort of membrane proteins helping this process out. So as a result, because it's being helped by certain proteins and your cell has a finite number of those proteins available, they might be saturated depending upon how much of the solute you're dealing with. Right? And there's an opportunity for competitive inhibition. Right? And the third would be active transport. So there are some cells that need to transport glucose against the concentration gradient. Okay? Certain ions, those same ions, the sodium, potassium, and calcium, need to actually be managed by the cell on purpose to keep these gradients built up. So the cell is constantly moving sodium from one side where it's low to the other side where it's high in order to just build up a concentration gradient that allows then the sodium to flow back through as part of facilitated diffusion. And importantly, the direction that these things are moving in is up against the electrochemical gradient. This would require some sort of energy provided by the cell to fuel the proteins that are going to help them out. And so, yes, it's going to require membrane proteins. And yes, that means there's going to be the potential for saturation kinetics and competitive inhibition. So don't be, overwhelmed by this table, because quite honestly, if you understand these concepts, then this is kind of like intuitive, all right? So rather than trying to memorize this as, okay, it's no, yes, no, 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 right? Understand it so that you can actually meaningfully be able to answer questions about this, right? Not just by remembering that in the lower right-hand corner, there were 3 yeses, right? But understand what those yeses are referring to. So let's look at the simple diffusion of gases. So oxygen, carbon dioxide, and water, they are able to diffuse across our cell membranes. Certainly, oxygen and carbon dioxide move a lot more readily than things like water. But if you think about it, right, the size of a water molecule isn't necessarily all that different than the size, right, if we're just talking about like oxygen, O2, or CO2, right? So it's more a matter of the fact that gases diffuse very easily because they are sort of neither hydrophobic nor hydrophilic. Right? So the diffusion of gases in and out of our red blood cells is the way that our red blood cells function. So in our human anatomy, our physiology, in our lungs, right, our lungs end with these different sacs that look like bunches of grape leaves. These are our alveoli, right? And each one of these is an air sac that the air coming in, that we're breathing in, has a chance to fill these air sacs. If you zoom in, here's one of these alveoli, right? The wall of the alveoli is super thin. It literally is just one cell layer thick. And right next to it would be a capillary, a blood vessel that its wall is also one cell layer thick. Right? So that means as you have things like your blood that's flowing through your veins, that gets pumped from your heart up into your lungs, the pulmonary arteries that pump into your lungs, right? You have your cells, the red blood cells that are flowing through there. The carbon dioxide that those cells are carrying from other places in your body just diffuse out, because the carbon dioxide is going from a higher concentration in your blood to a lower concentration in the air sac, the alveoli, and then that's the CO2 that you breathe out. At the same time, right, the oxygen inside the alveoli will just naturally diffuse from a higher concentration into the lower concentration, which is in your blood, and then that O2 can be bound up by the hemoglobin in your red blood cells, and the red blood cells then move on to deliver that to other places in your body. Right. So facilitated diffusion of red blood cells might involve something like the facilitated transport of glucose. So we have a particular transporter protein known as GLUT1, or the glucose transporter. Right. And so this protein takes glucose that's floating around on the outside of the cell, right, and it transports it through the membrane into the cell. And so this would typically be from high glucose on the outside to low glucose on the inside. Therefore, it doesn't require any energy on the part of the cell to do it, but the cell needed to provide this GLUT1 protein as sort of a channel, right, or a pathway for the glucose to get through the cell membrane, because normally the glucose molecule would not do that on its own. Okay. There are some examples of transporters, facilitated transporters, that don't just move one ion in one direction. Here's a fancy one that moves bicarbonate from inside of your cell, where there's a higher concentration, out where there's a lower concentration, while at the same time moving something like chloride ions, right, from a high concentration outside to the inside. So in other words, the shape of the protein is just right to do this trade, right, allowing one ion to move in one direction while the other moves in the other. So here is a cool image of the GLUT1 transporter protein here. So it's a protein that's made of 12 transmembrane chains, right? So it's just one peptide that kind of zigzags 12 times through the membrane, right? So of course, the amino acids that make up these transmembrane regions, you would imagine, are hydrophobic amino acids, right? So that the water molecules, look at this, this is cool, because normally in these figures, all of this stuff would just be like blank white on either side, right? But what are all these little messy speckles here? These little messy speckles are water molecules, right? So this helps you realize, yeah, this is how it normally really is. On either side of the membrane, right, there are all these water molecules, and it's the water molecules bumping into the hydrophobic parts of the GLUT1 protein that keep those transmembrane parts of the protein right in the membrane, right? So here's the lipid bilayer. So these green things are the phospholipids. And yeah, this is a nice reminder that you do have a few water molecules, right, that can make their way in between here, right? But obviously nothing compared to the water concentration on either side. And here's the glucose molecule. So here's the simple sugar that attaches to the side of the GLUT1, and then the GLUT1 is now going to change its shape so that it can move the glucose across to the other side of the membrane. So this cartoon depicts that change in shape. And so this is an important concept that I want you to keep in mind. Right? So here's the GLUT1 transporter. All right, so it's a protein that's embedded in the membrane. In the shape of the protein, it's kind of fluctuating. They draw it almost like a clothespin here. So you can imagine it that way, right? I mean, obviously it's not really a clothespin, but the point is, right, that it's a fluid shape that can change its overall configuration. So if the glucose transporter happens to be in this conformation and a glucose molecule, right, happens to get into it, so the shape of this GLUT1 transporter would be just the right shape to hang on to this glucose here. Once the glucose is in there, that kind of stabilizes or changes the shape of the GLUT1 protein, right, so that the GLUT1 protein, the opening, instead of being on the outside, then it opens towards the inside of the cell. And as a result, the glucose naturally, right, it's not actually covalently bonded in there. It was just sitting in there. So if this side opens up, then it could just fall out and go into the aqueous environment of the cell. And once again, the glucose transporter then is just kind of fluctuating, right? And if it happens to be in the open configuration on the outside, when the next glucose molecule comes, right, it'll repeat the same thing. Right? So in this case, right, the actual configuration of the GLUT1 transporter is just determined largely by where the concentration of the glucose is. If there's more glucose on the outside, then there will be a tendency for the GLUT1 transporter to naturally be caught in this conformation more because you have all the glucose molecules bumping up against it. Okay, so some carrier proteins can move things, like we just said, in one direction. So the GLUT1 transporter that we looked on the previous slide is what we call a uniporter. It moves one type of substrate, capital S here, in one direction, right, at a time. And it's quite possible that if the gradient changed here and the S was higher concentration on the inside, it's quite possible that it could then make the transporter go the other direction as well, right, and move it out. Other transporters, as we've already suggested, don't have to work on just one solute molecule at a time. It might work on two different ones. So this would be a different type of transporter that works on solute A and solute B. It is the right shape to bind both at the same time on one side, and then it changes shape and it releases both to the other side. Right? So because both the solutes are moving in the same direction, this is what we call a symporter. Okay? And we already saw that we have other transporters like the bicarbonate chloride one that can move substrates in opposite directions. Right? So again, amazingly, the shape of the protein is just right to be able to facilitate these types of movements. So the last slide I just wanted to wrap up with today is, let's look at the example of water again, because water is a really good example of simple diffusion and facilitated diffusion. Right? We said water molecules can certainly make their way through the lipid bilayer, but because the lipid bilayer is hydrophobic, it really slows down the ability of water to get across. Right? So certain cells of your body, for example, the cells of your nephrons in your kidneys, their job every day is to transport a lot of water, right? Because your kidneys are filtering your blood, right? So it's trying to recover a lot of water as it's then allowing waste products to be concentrated into your urine so your body can get rid of it, right? So these cells handle a lot more water than your typical cells do. So they cannot rely on water simply diffusing through the cell membrane nice and slowly, right? They need to be able to have certain channels that allow water to move in much more rapidly. So the diffusion of water is what we just call osmosis, right? And so certain cells, like kidney cells, have these special aquaporin proteins. That's what this gray thing is here. So this gray thing would be embedded in the membrane. And you can see it's the right shape, that it has a channel in the middle that allows water molecules to flow very quickly through them. But the amazing thing about it is it's exactly the right shaped channel so that at a certain point, the water molecules are forced to move in a single file, right, through there. I mean, that's pretty incredible if you think about it, right? But that's how the cell is able to control right? How much water is coming in. So this on the right is a stain of some tissue from the kidney. The blue stain, I think, is staining for the nuclei of cells. And what they've done is they've used this brown stain to actually see which cells are expressing a lot of these aquaporin proteins on their surface. And so you can see those cells that are expressing a lot of the aquaporin proteins are ones that are sort of clustered in these nephron regions. And this is where they're filtering the water. So with the help of these aquaporin proteins, right, these cells of your body are specialized. So in other words, as part of their differentiated cell function, they express the genes that code for these aquaporin proteins at a much higher level than other cells in your body that, of course, still have the same gene, but maybe just don't use it as much. Right? So how much water do your nephrons of your kidney cells deal with every day? Right? So a typical person's body has about what? Between 4 to 6 maybe liters of blood. Right? 6 is kind of on the high side. It's probably more like 4 to 5 liters of blood a day. Most of that is water. So how much actually gets handled by these cells in your kidneys? Astoundingly, our kidneys process around 180 liters of water every single day, right? That means they're constantly, that your blood is constantly going through them and being filtered. 180 liters of water being handled by the cells of your nephrons through these aquaporin proteins. That's like 92-liter bottles of soda every day that these cells are dealing with, right? And that's pretty amazing. And again, the thing I want you to think about, all the cells in your body have the same gene that would code for and make the aquaporin protein, right? And I think all the cells in your body do make it, right, to a certain degree. But your kidney cells, because their job is to handle so much water, right, because of their gene regulation, their differentiation pattern, right, they make a ton of these aquaporin proteins. So their surface is covered with these aquaporins so that they can handle their fully differentiated kidney cell job of handling this much water. Okay. All right, let's go ahead and stop there.